Today, the first day I had full voice, yesterday I had no voice, today I have half voice. I hope it lasts for 45 minutes, and I hope also that I make a very clear, straightforward presentation that you can also easily follow, but also more important that you can apply afterwards in your day-by-day work. So I think more and more, when we talk, as I mentioned on the first day in the course, when we talk about maturation and differentiation of cells, for the different cell types, we should not be restricted to one sample or type of sample, because maturation compartments and maturation pathways usually go on for different cell images in different places, and maybe particularly for cells that are related to the immune system, like the monocytes. So basically, if we look at the current concepts about monocytes and what you call the monocytic macrophage system, the very basic way of seeing it is that monocytes are produced in the bone marrow, they arrive into the blood, and from the blood they go into tissues. And from the tissues they go nowhere, because apparently no one knows where they go. At the same time, this is not that simple, because already in the tissues, macrophages from different tissues might have different appearance, different functionalities, so they are heterogeneous. But also in the blood, you can see different subsets of what we call classical and non-classical monocytes. So today we will be focusing mainly on the bone marrow and the blood compartments, and we will not touch the tissue side. As I mentioned 2 days ago, in the bone marrow, within the CD34 cells, CD64 is the kind of the marker that we can rely on to identify the earliest monocytic precursor. And if you remember, within the CD34 compartment, these cells expressing 64 that follow a maturation completely different in the beginning from the neutrophil 1, and you combine MPO and 64. These cells, in fact, represent around 6%, 5% of all the CD34 cells. After acquiring 64, you can follow the maturation, like with acquisition of myeloperoxidase, or with loss of markers of stem cells, like the stem cell factor receptor, c-KIT. So after this first dissection of the pathway, then it becomes very easy to follow a monocytic maturation in the bone marrow. So if we combine in one single tube markers that disappear or appear at different maturation stages, we are able to dissect the maturation of monocytes in the bone marrow. And as I mentioned also the other day, only part of this last stage of maturation, only part of it is what we call mature monocytes. So these compartments are the ones that we call morphologically under the microscope monoblasts, up to here, more or less, and promonocytes. So, in other words, by using these markers, you can make many more maturation stages than those that we can identify by morphology. And the basic reason is that these cells are not a major compartment in the bone marrow. So if you count 500 cells, you don't get many of them. You mainly get mature monocytes. So they are not easy to see if you don't analyze enough cells. So if you establish with these markers the maturation pathway in the bone marrow, you will realize that 64 is in fact the first marker to appear, and it's rather stable in the bone marrow through all the maturation stages until the mature monocyte. In parallel to this increasing 64, CD34, and maybe slightly later on, but very close, CD117, they go down. There is downregulation, early downregulation in a monoblast stage. So monoblasts can be 34 and 117 negative. There is downregulation of these two markers. HLA, by contrast, at these stages, you see, even might appear to increase slightly. However, HLA-DR decreases later on and gets up in the more mature compartments. So HLA-DR has a less stable expression during maturation, but interestingly, and as expected, it's always positive. It never becomes negative. It's just a question of how much molecules per cell. At the same time these markers disappear, there are markers that get upregulated. And these are the functional molecules for the monocyte. So usually these are receptors or proteins that you can link with specific functions for the monocytes. So among these molecules, CD36 would be after 64 the next one to appear. Then CD35, complement receptor. Then CD14, another molecule associated with the receptors in monocytes. And finally, the last one in this set of markers, CD300E. So from a practical point of view, if you would like to identify the mature monocytes in this pathway, you have to look at the very last stages where CD300E is highly expressed. Maybe the two last stages out of 20. For sure, you have to take into account that this is fluorochrome dependent. So if you change fluorochromes, maybe even it looks like the sequence might be different. Particularly when two markers come very closely one and the other. So if you change the fluorochromes of 14 and 35, then it might appear to you that 14 appears before 35. So if we make a summary, like we did yesterday, of the bone marrow maturation of cells from the monocytic lineage, this would be the summary. For sure, not all bone marrows are the same. They may show some variation. But in a normal bone marrow, the patterns of antigen expression, in general, there are always exceptions, in general, with a fixed panel of not only CDs, but also antibody clones, you can see that normal is normal. There is not a case that really goes out of what you would expect from a normal pattern. Also, interestingly, if you look at this line in gray, the number of cells per stage of maturation also goes up. So the more mature the cells are, the more cells you have from that stage. This is telling us that maturation goes along with expansion of the compartment. And for sure, for some maturation pathways, we think that division of cells occurs at a particular stage of maturation. Well, these curves, they are telling us that apparently it occurs all over the place at apparently very stable rates. Unless a maturation stage is not defining properly a maturation stage. But I would say it's the opposite. It's because we have defined maturation stages based not on multiple markers, but on a few. So if we define maturation stages based on 2 markers, then we might see big differences. So by using such a set of markers, you can explore what is the expression of other proteins on these maturation stages. And you can create, just for, let's say, practical purposes, you can create well-defined stages of maturation, 34 positive, 34 negative monoblast, raw monocyte, 14 negative, 14 positive, and mature. So you see that the numbers increase almost for all stages, especially if you look at the percentage within the maturation, except maybe down here. And why down here? Well, because those are the cells that should leave into the blood. So if they should leave, you should see only a small part. So by defining these maturation stages, then you can look at the expression of all those markers I was showing before. And green means negative. For instance, SLAM is a molecule that is not expressed at all in the bone marrow monocytic compartment. Or for instance, CD163 only maybe starts to appear at the latest stages, but the intensity even is not very high. You have to take care with CD163 because if you analyze samples with heparin, this marker doesn't stain properly. So, depending on the anticoagulant, that's the same thing for CD138. That if you use heparin anticoagulated samples, the stain is not as good as we did it here. And as you see, the latest marker to appear is CD300E. In fact, this is the marker that really can highlight the last stage of maturation. Maybe with CD312, but the differences here is from green to red, here is from, very dim expression, but basal expression to a slightly higher expression. Other markers like CD62L are pretty stable, CD33 pretty stable, cytoplasmic 68 pretty stable, 11C is progressively increasing in the maturation like CD45. CD13, like for the neutrophil maturation, has a high expression, then is downregulated in the middle, and then becomes upregulated again in the last stages of maturation. And the MPO, like for the neutrophil maturation, is higher in the beginning and lower at the end. So it's produced at relatively early stages, and after that, every time the cell divides, gets half.